You know that feeling when you're sure you've heard the same idea before, just dressed up in different words? Well, it turns out that happens in science. A lot. Today, we're going to look at this incredible paper by Mika Rubinov that's basically a decoder ring for complex science. It shows how a ton of methods we thought were totally different are, well, secretly, the same thing. It's time to find the simplicity hidden inside all that complexity.
So this is the big question, right? Are scientists just reinventing the wheel over and over again? I mean, the world of science is just exploding. New papers, new techniques, new algorithms are popping up constantly, and that's great. But it creates this huge problem. How do you even keep track of it all? More importantly, how do you know if some hot new method is actually new or if it's just an old idea wearing a fancy new hat?
And the scale of this problem is, well, it's just staggering. Just look at that number. Over 3 million scientific papers get published every single year. So yeah, it's easy to see how different fields can get totally flooded with methods that are basically doing the same thing. And the paper we're looking at today argues this isn't just inefficient, it's actually dangerous. It could lead to stagnation, right? Where we're all so busy creating new names for old ideas that we stop making actual, real breakthroughs.
Okay, so how do we possibly put through all that noise? Well, this paper proposes a really powerful solution. It's all built around a concept called analytical equivalence. And I love this. You can really think of it as a decoder ring. It helps us see the hidden language that all these different scientific tools are secretly speaking.
Now, the key phrase here is guaranteed by mathematics. This is so important. We're not just talking about two methods that happen to give you similar results. No, this is way stronger than that. It's a mathematical guarantee that, under certain conditions you can actually define, two different-looking methods are literally the same fundamental process. They just speak different languages.
And this really gets to the heart of it. Strong correlation is when two things tend to move together, right? The results look alike. But analytical equivalence is a whole other level. It means they are the same thing, just expressed differently. So you might have one method that speaks the language of data clustering and another that speaks the language of network science. But when you look under the hood, the actual math, the core calculation, is identical.
All right, so let's see this decoder ring in action. This first example from the paper is a huge one. It's this aha moment that uncovers a hidden link between two super popular methods. I'm talking tools that have been used in like tens of thousands of studies without anyone realizing they were related.
So on one side, you've got k-means clustering. This is a total workhorse in machine learning for just sorting data points into groups. And on the other side, you have modularity maximization. This is a cornerstone of network science for finding communities, you know, those dense clusters inside a network. They look different, they sound different, they come from totally different worlds. But at the end of the day, they're both trying to do the exact same thing, find meaningful groups in the data.
So how can these two giants be the same? Well, the secret isn't some crazy new math. The paper puts it perfectly. It's all about what you do to the data before you even run the algorithm. The core ideas were basically identical all along. It's like we had two identical engines, but we were just feeding them slightly different kinds of fuel without realizing it.
So how do you actually turn one into the other? It's surprisingly simple, just a couple of tweaks. First, you take a part of the modularity formula, and you stop thinking of it as some complex statistical thing. Instead, you treat it like a simple data cleaning step. Think of it like removing the background hum from an audio recording. And second, you just adjust each group score based on how big it is. That's it. You make those two little changes, and boom, the modularity formula is the k-means formula. It was the same engine under a different hood the whole time. And what you end up with is this beautiful unified framework. K-means and modularity, they're not these completely separate things after all. They're just different points on a spectrum. The only thing that separates them are those two simple data prep steps we just talked about, whether or not you do that baseline removal and whether or not you adjust for the size of the group.
Okay, so this isn't just about clustering. That was just the start. The paper goes on to show how this way of thinking, this decoder ring, can simplify all kinds of super complex, multi-step processes, especially in a field like neuroscience.
Okay, let's take a look at this. Say you want to find what are called coactivity gradients in the brain. Basically, the main patterns of how different brain regions work together. The old way of doing this was, well, it was this super convoluted 7-step process. It used a fancy technique called diffusion map embedding, which is powerful for sure, but it's also kind of a black box. You put data in, you get an answer out, but it's hard to know what's really happening inside.
But here's the kicker. The paper shows that whole complex, 7-step black box process is mathematically the same as doing something much, much simpler, just finding the main patterns in a basic shared neighbor network. And how similar are the results? They correlate at 0.988. I mean, that's practically identical. But one way is vastly simpler to calculate and, more importantly, to actually understand.
And here's another great example. A lot of you have probably heard of UMAP. It's a super popular tool for visualizing really complex data. But let's be honest, how it works under the hood is pretty complicated. So the paper introduces this new version, UMAP, which is a much simpler take on it, built on those same ideas from modularity. And what's cool is it connects UMAP back to older, more intuitive methods, the kind that work like you're arranging dots connected by invisible springs.
Now, you might think, okay, a simpler version probably means you lose some quality. But get this, for the brain imaging data they used in the paper, this simpler approach, it actually produced visualizations with about 8% higher fidelity. Better results from a simpler method. It just goes to show you, sometimes simpler really is better. So, when you put all these examples together, you start to see that this is about more than just cleaning up our scientific toolbox. This is about changing how we think about scientific progress. It's a shift away from just chasing what's new and fancy and towards a focus on what's clear and what's true.
And here is maybe the most striking example of this redundancy. On one side, you have average controllability. I mean, that sounds super sophisticated, right? It's a fancy measure from network control theory. On the other side, second degree, a super simple, basic statistic about a network structure. And it turns out they are almost perfectly correlated. We're talking an R value of 0.999. For all practical purposes, they're the same measure. It's just that one comes with a whole lot more theoretical baggage.
And this all points to a really fundamental shift in how we should be thinking. The paper argues that we spend way too much time debating which fancy algorithm to use, when really we should be spending our energy justifying the choices we make about the data before it even gets to the algorithm. In other words, the choices you make about how to frame the problem are way more important than the black box you throw the problem into.
And what's really cool is that this perspective can help settle some really long-standing debates in science. Take neuroscience, for example. There's been this huge argument for years about how the brain is organized. Are there distinct networks of regions, or is it all just smooth gradients of activity? Well, this paper shows that you can make the two views mathematically equivalent. The biggest difference is just a simple data transformation. It turns out the two sides weren't arguing about what the brain was doing. They were just wearing different glasses.
Okay, so let's bring this all home. Let's really boil it down to the key takeaways and talk about why this way of thinking is so important for the future of science.
So, takeaway number one, a lot of the time, complexity is just an illusion. That hot new method with a super complicated name, there's a good chance it's just a simpler, familiar tool that's been applied to data that got tweaked a little bit beforehand.
Takeaway number two, we need to shift our focus. Instead of arguing endlessly about which black box algorithm is the best, we should be asking why we're transforming our data in the first place. Why did I remove that signal? Why did I choose this specific way to measure similarity? That's where the real science is happening. And finally, takeaway number three. Finding these hidden connections is how we accelerate progress. By actively looking for these unifications, we can stop reinventing the wheel. We can clear out all the clutter and focus our energy on what actually matters, solving the next big problem.
And that leaves us with a really big, exciting question to think about. This whole decoder ring idea. It's not just for neuroscience or machine learning. So I want you to think about this. What happens when we start applying this lens to other complex fields? Like economics or genetics or even social sciences. How many competing theories out there are actually just different ways of saying the same thing? Finding those hidden connections, well, that could be where the next huge breakthroughs are waiting.